Start our next lecture, which is DNA repair. All right, so we said that DNA sequence contains information, but DNA is a chemical molecule, so it can have damage. Okay, so and these damages, which we call mutations, these damages can lead to changes in the genome sequence, which we call mutations. Okay, so these mutations can be caused by damage that is coming from the environment, or due to the process of DNA polymerase making errors during replication. So mutation, basically, we call it a change in the nucleotide sequence of the genome. There are different kinds of mutations, and this is some of the terminology that you will hear. Point mutations are those that occur only one nucleotide change. We will talk about insertions or deletions. These will be basically one or few nucleotides. There are also larger mutations in the genome. These can be large insertions and duplications and deletions of parts of chromosomes or whole chromosomes. Okay, so that's the type of mutations that usually you see in the genome. All right, so these are caused due to errors during replication or the damaging agents of mutagens. And these mutagens could be chemicals or radiation or heat, etc. All right, so what's another mechanism that can cause larger mutations in the genome? In addition to mutagens like chemicals, etc., and replication errors. Give me one mechanism by which you can also have mutations in the genome. I know some of you are here for lecture 10. Think hard. Mutations are changes in the sequence of the genome. Go ahead. Could it be some sort of virus? You're on the right track. Yes? Did you? No? I thought you hold your hand up. No? Okay. Go ahead. Transposons. We talked like half a lecture, guys. And we said transposons cause a lot of mutations because they jump around in the genome and they can cause problems, right? So transposons is another one. All right, so the damage to DNA is extensive. So the estimates of DNA damage per cell changes in the range of 10,000 to 1 million a day. So this is per cell, right? So there's a lot of damage to cells. And some of this is basically due to inherent instability of the DNA because the bond connecting a purine base to the deoxyribose is actually prone to hydrolysis at a low rate under physiological conditions. So that's just because DNA is basically a molecule. And you can also have damage from normal cellular reactions that produce hydroxy radicals or superoxide. And one such reaction occurs in the mitochondria because during the ATP production, there's some reactive oxygen species that are basically leaks out as a byproduct. Okay? Some of these species, as first line of defense, are converted back to non-mutagenic chemicals, for example, by an enzyme called superoxide dismutase. Basically takes that oxygen radical and then converts it into regular oxygen on the hydrogen peroxide, and that's the first line of defense. And these are not as mutagenic. All right, so you can think of like cells having mechanisms to say, okay, not so fast hydroxy radical, right? So there are defenses before the chemicals can turn into mutagens. But there are lots of other damaging agents, and here is a table that is listing the type of damaging agents, the type of damages that they cause, and the type of DNA repair mechanisms that the cells use to fix these damages, okay? So, for example, ionizing radiation, X-rays from the sun, or anti-tumor drugs, For example, they can cause double-strand breaks, single-strand breaks, or they can cause cross-links in between nucleotides. And these type of DNA lesions, these type of DNA damage are repaired by two pathways. One is non-homologous end joining, and the other one is homologous end joining. And there's another column telling you the fidelity of the pathway. Fidelity means how well does it restore the information back to its original sequence. Okay, so how well the repair works, how good is it? So non-homologous end joining doesn't do very well. Homologous recombination is pretty good, and some of the other repair pathways are much better. All right, so UV light and some chemicals can cause pyrimidine dimers. So these are thymidine dimers. This also can cause addition of bulky adducts. Basically what that means is add some molecules to the base itself. And these are typically repaired by nucleotide excision repair. Some oxygen radicals and spontaneous hydrolysis, some alkylating agents can also cause damage to the base. They can cause abasic sites. That means the sugar and the sugar phosphate backbone stays intact, but the base is taken out, right? So ATGC is taken out from that site. So these are called abasic sites. They can cause single-strand breaks or have a version of guanine, which can cause creation of a version of guanine called 8-oxoguanine. And these are repaired by base excision repair. And then you have replication errors due to DNA polymerases not being perfect. Okay, so these can cause mismatches or insertion of a different a chemically different nucleotide, or it could be skipping of a nucleotide addition that causes deletions, or addition of extra nucleotides can cause insertions. And most of these replication errors are fixed by a replication coupled DNA repair system called mismatch repair. All right, so this is kind of the summary of what we're going to talk about in this lecture. So we said there are different types of damages, spontaneous errors and mutagens, and these can cause a variety of different damages. These could be mismatch, addition of adducts, etc., or double-strand breaks, etc. So let's start with these small damages to the DNA. Okay, so the first cause of damage is the DNA polymerase, which can incorporate the wrong base during the replication. So DNA polymerases usually makes mistakes at a rate of about 1 every 100,000 nucleotides. These are different in between different species, but approximately that much. That's a lot, because imagine that we have 6 billion bases in each diploid cell, so every division we should be accumulating 120,000 such mistakes, right? So that's a lot. But 99% of these are fixed by the proofreading activity of the DNA polymerase. So how that occurs is like this. So you have DNA polymerase. It has the template strand here shown in blue. It has the new strand here shown in red. So normally what it would do is it would take the template strand and add the correct base complementary to the nucleotide here, right? But sometimes if it incorporates the incorrect base, what happens is that base pairing creates a structure that's not normal, and that the abnormal structure is sensed by the polymerase itself. Imagine like your hand is used to holding the nucleotides like this, and you have an incorrect nucleotide sticking into your hand says kind of reacts, right? It's kind of like that, where the polymerase structure does not like that incorrect incorporation. And what it does, it goes through a conformational change and pushes that newly synthesized strand into a different domain of the DNA polymerase that contains an exonuclease activity. And what that exonuclease activity does is it removes this last nucleotide that was incorrectly incorporated, and then pushes back into the normal configuration of the polymerase, and now it can insert the correct base and continue its business. Okay, so this is basically proofreading activity. But the proofreading is also not perfect, so leaving about 1,000 or so mistakes to be fixed by another DNA repair mechanism called mismatch repair. So this mismatch excision repair machinery repairs mismatches during replication. They go along with the replication machinery, checking that the newly replicated DNA is replicated correctly. So you have two proteins here, MSH2 and MSH6. They can recognize, for example, the incorporation of an incorrect base. So this is the template strand up the top. You have a newly synthesized daughter strand, which has incorporated a C compared to A, which should have incorporated a T, right? So this causes a bulge in the DNA structure, and that bulge is basically recognized by these proteins MSH2 and MSH6. And these proteins, because they ride along with the replication machinery, they have evolved strategies to recognize which strand is the newly synthesized strand versus which one is the template strand. Okay? So these proteins, when they bind, they know that this C is the incorrect base. And they recruit these proteins called MLH1 and PMS2, which are endonucleases. Endonucleases are those proteins that can cut the DNA sugar phosphate backbone inside. Okay? Exonucleases are the ones that can cut from the end. Endonucleases can cut from inside. What's the endonuclease enzyme that you can think of from your molecular cloning project? Molecular cloning lecture. Restriction enzymes are endonucleases, right? They cut right into the sugar phosphate backbone. So these endonucleases cut right here, and right here, okay? At a stereotypic distance that is specific to these endonucleases. And then an exonuclease chews the end of chews out the end of the DNA by the help of a helicase, which opens up the strands. So the strand is opening up, and the exonuclease is chewing this off. And what you will have is basically a gap right here. And that gap is recognized by cellular DNA polymerases. And what they will do is they will just take this as a primer and complete the synthesis of this. And a ligase will basically ligate the end. And you're going to have the correct base inserted during this replication process. Okay? So the mismatch repair enzymes will basically just remove the part of the newly synthesized strand containing the mistake and re-synthesize it with a new DNA polymerase. Make sense? Okay. So these processes are really important because if you don't correct a lot of these mistakes, it can cause mutations. And in fact, for example, mutations in the MLH1 or MSH2 proteins themselves actually predisposes people to colon cancer. So the more mutations you have, the likelihood that you will have cancer. All right. So there are other damages, and one of the damage that is another very common damage that occurs is deamination of the methylcytosine, which can lead to a C to T change. Okay? In humans and a lot of mammals, most of our cytosine nucleotides are actually modified by the addition of a methyl group. So we have a lot of 5-methylcytosine in our DNA. So it is addition of a methyl group in the 5th carbon, right? And then if that 5-methylcytosine gets deaminated, so that removal of this amine group here, it basically turns into a thymine, okay? So that 5-methylcytosine can turn into a thymine if it loses that amine group. And this can occur spontaneously, or it could occur due to some enzymes doing this, actually. So normally in your normal DNA, you will have a methylated cytosine that's base pairing with a guanine. And then if you get deaminated, then you turn this into a T. If this mistake is caught by another repair machinery, which is called base excision repair machinery, it will revert this back to a C, right? And then you will be back to normal. But if this if this damage is not repaired before the next round of replication, the DNA polymerase, what it will do is it's going to incorporate an A as a complementary base against the T, and then the other strand is going to take the correct nucleotide. But what you'll have is you'll have one sister chromatid containing a mutation, which is basically a C to T change, and the other sister chromatid will have the correct sequence. Does that make sense? All right. So how does base excision repair work? So for base excision repairs, there's some proteins that go along the DNA and look for damage. The type of damage they recognize can be mismatches. It could be addition of adducts to the base. It could be regions of the DNA where the base is missing, so abasic sites. And once it recognizes these sites, the first thing that it does is to decide which one is the correct strand, right? So to a protein that's not traveling with the replication proteins, it doesn't know which one is the right strand. But throughout millions of years of evolution, there are proteins that have evolved to assume that this T to G mismatch is due to a C methylated cytosine turning into a T, right? And now what it does is it assumes that this is the wrong nucleotide. So it comes in, and the first thing that it does is it removes the base. So it removes the T base, leaving an A basic site. Okay, so this is an intact sugar phosphate backbone, but it's missing the base. And then this structure recruits another endonuclease, and what it does, it cuts the sugar phosphate backbone on one side, and this recruits additional proteins like AP lyase, which also removes the damaged nucleotide, A basic nucleotide completely. And then this gap is repaired by a specialized DNA polymerase called DNA polymerase beta, which adds the correct nucleotide, and the ligase basically ligates the phosphodiester bond. Okay, so that's how this can be repaired. So there are other DNA glycosylases that are specific for other sets of damages. So, for example, there is a DNA glycosylase that is specific to recognition of 8-oxyguanine. This is basically an oxidized form of guanine. And so also depurination, which means just loss of G or A from the DNA, also is repaired by base excision repair. Any questions about this? All right. Other damages. This looks so good. Is from the sun, right? So you have UV rays that are coming from the sun, and that also can cause DNA damage. And the typical way the UV rays irradiation causes DNA damage is that it causes a chemical bond between thymine and thymine, two thymine nucleotides that are next to each other in the DNA. Okay, so they cause the thymine-thymine dimers. All right, so again, this is not so great for the shape of the DNA, right? The molecular shape of the DNA does not want any thymine-thymine chemical dimers, and what happens is that this shape changes, and you have these thymine-thymine dimers bulging out into the DNA. outside, just out the DNA. And that shape is again recognized by proteins that go around and do a surveillance of the genome and recognize that there's a TT dimer here. And these proteins are 23B and XPC. So what they do is they recognize these damages and they bind to it and they start changing the conformation of the DNA to start opening up the DNA double helix. And then they also recruit other proteins. And these proteins are RPA, TF2H, and XPG. And what they do is TF2H has a helicase activity. You can remember it from TF2H, H for helicase. Opens up the DNA molecule here, about 25 bases or so, and then recruits some other proteins. They also recruit these two endonucleases, XPF and XPG, and they basically cut the strand containing the TT dimer to remove that DNA that's containing the damage. Okay, so that basically gets removed out. And it gets chewed out and gets the cell gets rid of it. But then again, that leaves a gap, basically, that will be recognized by the cellular DNA polymerases and the DNA ligase that will newly synthesize the DNA that's complementary to the template and repair the problem. Okay? Any question about this? All right, here's a question for you guys. Mutations to which parts of the genome would have a larger impact? We've learned about what type of sequences are in our genome based on that table from the lecture 10. Think of what parts of the genomes, mutations to which parts of the genome would cause the biggest problem. It's not that hard. Coding sequences, right? If you make a change in the DNA sequence, cause insertion deletions, you're going to change the protein sequence, and that's going to change the protein structure, and that's going to change the protein function. What other places in the genome could cause problems? Regulatory regions, definitely. Introns or the places that cause splicing problems, etc., right? All of those functional regions. In fact, these transcribed regions are the places that should be constantly repaired, because if the repair stays during transcription, then you're going to make the wrong protein, for example, right? And what's interesting is that this TF2H protein is a general transcription factor. That's why it's called TF, transcription factor. That has a helicase activity, which also works during transcription initiation. Okay? So, in fact, this protein can couple DNA repair to transcription so that you repair the transcribed regions in a lot more frequency than regions that are not transcribed. For example, some repeat region, who cares if it has another mutation in it, right? So that's how that can be skewed. So a lot of these genes involved in DNA repairs have been isolated from genetic experiments that were done in model organisms like yeast, right? And some of them in humans have been identified by looking at rare genetic diseases where people are sensitive to damage. One of these genetic diseases is I don't know how to pronounce that, Xeroderma pigmentosum, XP. These happen in people who cannot tolerate UV light. They cannot tolerate sun at all. So these children are also colloquially called the moon children. So they have mutations in the genes, in some of these genes, nucleotide excision repair genes, that are important for functions during that nucleotide excision repair pathway. All right, so that was mismatches, nicks, and adducts, those small little things that can happen to DNA. And there are other damages that can happen to DNA that could cause double-strand breaks. And these are probably the most dangerous types of DNA damage because the repair of them is prone to errors. Okay, so they have low fidelity. All right, so when you have a nick inside the DNA, so that's just a little cut in the phosphodiester bond. If the phosphodiester bond is broken, but there's no damage to the 5' phosphate or 3' hydroxy groups, what will happen is the DNA ligase will recognize this nick, simply patch it up, and then the nick is repaired. Easy peasy, right? But if this nick is not repaired before a replication fork arrives, what will happen is that, imagine, so this is the leading strand that's being synthesized and coming up here. It goes down here and comes all the way to the nick. And all of a sudden, you basically broke the replication fork because you no longer have a template, right? In these cases, what we call the replication fork collapses. What that means is it just stops, okay? So because it has nowhere to go. When that happens, this can be repaired by homologous recombination. I'm sure you love homologous recombination by now. So what happens is that you have exonucleases that is going to chew out one end of the double-stranded DNA that's containing the break here, leaving out a 3' strand here that is a single strand, which can look for homology. And it's going to find homology in the sister chromatid. It's going to invade the sister chromatid. And it will start, the DNA polymerase is going to start adding nucleotides, and the helicase is going to open up. And what will happen is that you're going to create a holiday structure behind the fork. And that holiday structure, when it's resolved, will create two nicks in the two sister chromatids. They can be ligated, and then this is basically the structure of a normal replication fork again, so that the normal replication fork can continue replicating. This is the leading strand again, and this is going to be the lagging strand. Okay? Make sense? Questions? All right. You can also have double strand breaks if the break happens in both phosphodiester bonds on both strands of the DNA, right? So this is what we call a double strand break. Both strands are broken, and they're very close to each other, leaving this DNA ends exposed. This type of break can be repaired in two ways. One is through homologous recombination, and the other one is called non-homologous end joining. And in this picture, we're going to go through how the homologous recombination will repair this. And it's essentially exactly the same thing that you guys already learned, right? So you have a double-stranded break that has that's here, and what you're going to have is you're going to have exonucleases that are going to chew up one end of the DNA, leaving 2 3' ends, single-stranded ends open. And these DNA are going to look for homology. So they're going to look for sequences that are complementary to here, right? And they're going to find those sequences in the sister chromatid. They're going to invade the sister chromatid and start extending. And that extension is going to continue until you have formed a holiday junction by ligating these two ends. And then the holiday junction is going to be resolved, leaving you with two intact sister chromatids. So this is basically homologous recombination that occurs due to having a double-stranded break. All right. At what cell cycle stage can this type of repair likely to happen? G1? Mitosis? Which one? Interphase. Interphase, yes. Which phase of the interphase this would happen? S phase, when the sister chromatids are right next to each other because cohesin is holding them together, right? You're right next to who you're going to copy from, not in the midterm. Don't sit next to people you're going to copy from. But that's essentially it, right? You have two sister chromatids next to each other, so the one sister chromatid with the break can copy the DNA from the other sister chromatid. When there is no template to copy from, when there is no DNA sequence to copy from, these 3' ends can look for homology elsewhere. Okay? So the homologous recombination can occur outside S phase, although at a lower frequency, because these 3' and single-strand ends are going to look for homology. And this is a problem for regions of the genome which have repeats, because you might not find the correct answer, you might not go find the correct homology in the sister chromatid, you might go elsewhere in the genome, right, and the wrong place. And that could happen especially because of the fact that 50% of our genome are repeats, right? So because these repeats, if they're broken, they can cause problems. What kind of problems they can cause? A lot of different things you can imagine. Here's just one example, okay? So imagine that there are two sequences that are repeated. These repeat sequences, they're not always exactly identical. They're sometimes a little different. So they have two similar but not exactly identical repeated sequences in the same chromosome. Let's say that you have a double strand break right here in the pink one, okay? And you created ends here that are going to look for homology. So you have a double stranded break, and these ends are going to look for homology. If the sister chromatid is not there, where it can find that homology, sometimes these DNA will find the homology in the other sequence that is repeated, okay? And then that could form structures like this. And if the ends invade the other repeat, sometimes because the sequences are not exactly the same, you're going to have mismatches, right? And these mismatches will abort the pairing and prevent recombination, because recombination proteins look for perfect homology. And when there is not perfect homology, they'll say something is wrong, let's just give up, okay? But sometimes they won't say that because either the pairing is perfect or that there is some error. So what will happen is then you're going to recombine this part of the chromosome to this part of the chromosome, and if the holiday junction resolves in a way that will join these together, you will be releasing a circle containing the DNA in between, okay? So the two ends, during the recombination process, the broken end and the template end can get joined and leaving the DNA in between out. And that's a problem, right? What is this going to cause? It's going to cause a large deletion, right? So it's going to cause a large deletion. So double strand breaks at the repeats is dangerous, right? So we don't want that happening. And in fact, something that we talked about in the last lecture, which is the heterochromatin structure, protects these repeats from homologous recombination. So we said that in active open chromatin, you have lots of acetylation. The nucleosomes are like feet on a string. They're kind of loose and happy. And in the heterochromatic regions, we have a lot of hypoacetylation, so less acetylation. You have inactive, that means not much transcription, not much going on, and condensed chromatin. And this structure are mostly found in repeat regions of the genomes. Give me an example. What region of the genomes with repeats contain heterochromatin?
The telomeres.
Telomeres. What else? Centromeres, right? So these regions have contained heterochromatin. So can you guys just think about how can this heterochromatin prevent homologous recombination?
So it will prevent like strand invasion?
Exactly. That's one of the mechanisms by which it will prevent homologous recombination. Imagine that you actually got a double-stranded break over here, okay? what needs to happen is that these nucleosomes need to be removed so that the exonucleases can come in and chew up, right, and release these 3' ends to look for places to invade. So not only you have to remove these nucleosomes, they have to come out and then also look for homology in the sister chromatid or some other part, right, which is also heterochromatic. So it won't be able to invade the DNA when the DNA is wrapped around the nucleosomes, right? So that's going to actually repress the homologous recombination machinery. Does this make sense? Because to have access to DNA, to be able to open up the strands, you have to take the DNA, unwrap the DNA off of the histones. You have to unravel the nucleosomes. You have to open the nucleosomes. And that's going to be hard when it's densely packed like that. All right. So the other way that you can repair double-strand breaks, like in the repeats or other regions, is a very error-prone way called non-homologous end joining. So let's imagine that there is a double-strand break, and it's not a clean double-strand break. That means that there's some bases or some the 3' phosphate, etc., are removed from the ends, right? So the break happened that it removed a few nucleotides or bases from the ends. So it's not blunt. So when that happens, these double-strand breaks can be repaired by the non-homologous end joining by the action of these proteins called DNA PK and the Q80, Q70 heterodimer. And what they do is they recognize these breaks and they bind to it and to hold the two ends together. Okay? But then what they do is they recruit some other proteins and they let them chew the ends so that you create a blunt end. So what they do is they bring proteins that are going to cut this and cut this, so you have a perfectly blunt end, right? So this is blunt. So you have two DNA with blunt ends, and then it's going to bring DNA ligase that's going to join it. What kind of mutation is this process going to cause? A small deletion, right? A few nucleotides are going to be deleted here because of this. Non-homologous end joining is particularly bad. Okay, so here's a question for you. We talked about replication errors causing both nick and double-strand breaks. Which is more likely to cause genome rearrangement? That means part of the genome, part of the chromosome goes on and sticks to another part of the chromosome. Will nicks or double-strand breaks cause bigger problems? Double-strand breaks, right? The reason is that if you don't have homologous recombination to repair that, non-homologous end joining can join the ends of the DNA. Normally, normally the two ends would be near each other. Okay, so when the double-strand break occurs, the two ends are near each other. But sometimes, if these two ends are separated, okay, so you have a part of the chromosome here and part of the chromosome here, right? And you have a double-strand break. So normally this will be close to each other. But let's say there is another chromosome here that by chance has another double-strand break here. The non-homologous end joining machinery actually does not know that this end needs to be ligated to this end, right? So that's, let's say, chromosome 1 ends. And then what it will do is it accidentally will ligate it to another end that it found in the vicinity, okay? Whatever is close by. And what that will do is it will join these two different chromosomes together. Make sense? And we know that this can occur because you can actually see it in the karyotype of some people with chromosome translocation problems. We call these translocations because some part of the chromosome is translocated to another chromosome. All right, so an example of this is the Philadelphia chromosome, which is associated with chronic myelogenous leukemia. So what you have is you have a normal chromosome 9, it looks like this, right? And then you have a normal chromosome 22, it looks like this. But in the Philadelphia chromosome, what you have is part of the chromosome 22 is ligated to the end of the chromosome 9, and that little part of chromosome 9 is ligated to the end of chromosome 22, causing this very long chromosome 9 with a part of the chromosome 22 and a very small chromosome 22. So these kind of translocations also can cause cancer. And they are mostly rise due to non-homologous end joining of double-strand breaks. All right. Repair is really important, right? And I gave you some examples if the humans have mutations in the repair proteins themselves, mutations in the genes that are encoding for the proteins, repair proteins themselves, this can cause a lot of hereditary diseases, right? And we talked about this skin disease because it's caused by mutation in the nucleotide excision repair. And then there are also other syndromes like Bloom syndrome or disposition to breast cancer because BRCA1 and BRCA2 are actually also homologous. They are involved in homologous recombination repair. Okay? So mutations in, this is pretty famous, right? Like the mutation in these two genes actually can cause breast cancer. Right? So a lot of these mutations in the repair genes are no good. And there is a link between mutations and cancers. In fact, some of tumor suppressor genes. So tumor suppressor genes are genes when mutated causes tumors. Okay, some of these tumor suppressor genes are proteins that are involved in the repair of the DNA damage. Okay, so you can see again there's a lot of correlation between mutations and cancer. All right, so for hundreds of years, people actually have been suspecting that mutations are linked to cancers because mutagens can cause DNA damage and that can lead to cancer. So as early as in the 1700s, people recognized that environmental poisons can cause cancer. They were studying chimney sweepers, for example, and finding a lot of cancers in these people because they're constantly inhaling bad stuff, right? And they've recognized that snuffing tobacco was associated with nasal cancer. And then in World War II, people realized very immediately that the leukemia formed in people exposed to radiation, right? So these are very strong correlations, and the correlations are good to look for because they can be a sign of causation, but not necessarily, right? There's a great website where you can actually look at spurious correlations. What you can see is the things that correlate really well, but makes no sense whatsoever, right? So for example, here in this plot, what the guy is plotting is that all the data collected at different years here, so 2000 to 2009, and he's plotting the divorce rate in Maine against the margarine consumption, right? And you can see that they correlate really, really well, right? But I'm pretty sure that eating margarine is not causing these guys to divorce, right? So the correlations can be really good, but they might not mean anything. So then you have to build some causation. So here is a couple of slides explaining how some of the researchers actually linked cigarettes to cancer. Okay? So one of the first observations that were made was that people who smoked had lung cancers and bladder cancers a lot more than people who didn't, right? A pretty good correlation. And in fact, if you look from 1900s to 2000s, the per capita cigarette consumption that's plotted in black here, what you see is that in 1950s, cigarette consumption goes up a lot because there's lots of lots of advertisements. It's so fashionable to smoke, right? And then after that, there was a campaign to reduce smoking, and the consumption goes down. And you can see that after a lag period, which are these people smoking for a while, they start developing a lot of cancers, right? And then with the the reduced use of cigarettes, the cancer rate also goes down. So these were males because the men started smoking earlier than a lot of the women. The woman caught up in 1970s, thinking smoking is awesome, and then what happened is, again, they started getting lung cancer, right? It's a pretty bad and pretty impressive correlation. And some of the people who studied this and found these correlations found that cigarette smoking caused cancer, and the first thing that they did is they switched to pipes, right? Because that makes perfect sense. But then they started doing more experiments, and they figured out, okay, pipes aren't good either. So some of the observations that have made. The first thing that they found is that 60% of the cancers that they saw in the smokers contained a mutation in the p53 gene. This is a tumor suppressor gene called p53. And at several p50 mutational hotspots common to all cancers. Hotspots are basically residues in the proteins that you see mutations over and over and over again. So you look at one patient, you see residue 128 mutated. You look at another person, the same residue is mutated. Another person, same residue. These are called the mutational hotspots. So they found these hotspots common to all cancers, such as in codons 248 and 273, but there was something curious. In the cancers of people who were smoking, they found that these mutations were usually a G to T conversion, okay? But in cancers that were not related to smoking, the same sites were containing a G to A mutation. That's telling you that the mutagen that's causing these changes are different in people who are smoking versus people not smoking, right? So that's the first clue that they got. And then the second clue came in from doing some experiments. One of the things that they suspected were causing the mutations was this compound that you find in cigarette smoke. It's called benzopyrene. And this benzopyrene can be converted to a much more mutagenic compound in the lung by our regular cellular enzymes. So they get converted to benzopyrene to a carcinogen, and that carcinogen becomes an adduct. So it's basically a chemical that attaches itself to guanine and causing this problem, right? So what the researchers did is they took some cells, the lung cells, bronchial cells that you can grow in a dish, and they incubated them, added some benzopyrene to these cells, and then incubated them for a while, grew the cells for a while, then took the cells, isolated the DNA, and then sequenced the p53 gene sequence. And then interestingly, what they saw was the same codons that you find in people with cancers were also changing the same way in these cells. So basically, the carcinogen was leaving its fingerprint in the scene of the crime, right? So then the researchers concluded that it is this compound that is causing the specific mutations in the tumor suppressor gene p53. And if you don't have that tumor suppressor gene functioning normally, then you start developing cancers. And because these things are due to inhaling and conversion of that benzopyrene to a more mutagenic compound by the lung cells using their regular own enzymes, you see a lot more incidence of lung cancer in people that smoke, right? Any questions about this?
How does the mutation in guanine change it to thymine?
This is repaired by base excision repair, right? Because it's a big adduct. But I don't know what in the process of that repair or the lack of repair causes that specific change. So it's probably due to some enzyme that is trying to repair this, repairing it incorrectly, or not repairing it and then getting another base that is complementary to it. So we would have to look and see structurally what's different.
Also, what causes the benzylpyrene to target specifically P53? How does it?
It doesn't. It targets everywhere, but P53, when it's damaged, then you get cancer, right? We're constantly having mutations and damages in our cells all the time. Most of them, who cares? They don't do anything, right? But some of them, if it occurs in critical genes, then they cause cancer. Right? Does this make sense? Good questions. All right. So now people switch from pipes to e-cigarettes, right? Because that makes sense. And especially because there's a lot of advertisements and these beautiful colors and etc. that's targeting to middle school kids so they can start smoking, which is awful. And the intention of these tobacco companies is pretty clear, right? What they have done constantly by advertising to women, advertising to young people in poorer countries. And because of that, now, yes, in the US we have a lot less smoking and a lot less cancer, but in developing countries this is still a really big problem because a lot of children start smoking very early. Due to this very heavy targeting. And in fact, in up to 2000, cigarette companies were giving out free cigarettes to young people in all these developing countries, right? So whenever you see headlines, e-cigarettes are awesome, they don't cause anything, just be a little bit suspicious about that. There has to be some science behind it to show that that's actually true. And hopefully not sponsored by the cigarette companies themselves. In addition, cigarettes are not only associated with cancers, they're associated with other diseases like heart disease, etc. And people haven't looked at it rigorously enough to see if e-cigarettes are causing these things or not, right? So if you're an e-cigarette user, I would suggest you also quit. I know it's hard, but it's better to quit. All right. Just like the chemicals in the cigarettes, there are other chemicals that get modified by the cellular enzymes, making them a more mutagenic compound, causing mutations. And for example, one example of this from your textbook is aflatoxin B, which is a fungal metabolite that you find in mold grains. What happens is that when that goes into the liver cells contain some specific proteins that are important for normal liver function, but they also can take this compound and change it into another compound that is very mutagenic and can attach it into guanosine again. Again, this is a large adduct that would be normally repaired by base excision repair, but if there is a problem in the repair process, then it could cause mutations. And that is associated with liver cancer. Another one that is really unfortunate, because I like eating meat, is the meat, the charcoal that you will get from grilling, right, at high temperature. So you cook meat at high temperature, and that causes the release of another chemical that can go into your diet and turn it in your diet, that can go into your cells and turn it into a bad mutagen and attach itself to the DNA. So there are lots to think about. All right. But one thing that I want to tell you also is the mutations are not always bad, right? Most of them, they don't cause any problem. Some of them, they do cause problems and associated with cancers. But over millions of years, if there was no mutations, you know how we would all look like? We would all look like exactly like each other, right? It would be an awful world. Imagine everybody looking the same, right? So mutations are basically create diversity in the phenotypes. And that is really important because that's important for natural selection, right? And that's important for evolution. And for example, if DNA repair were perfect and there were no mutations in the populations, then there would be no genetic variation whatsoever. And without genetic variation, natural selection would not happen. It doesn't have anything to work on, right? So we should have enough mistakes for evolutionary mechanisms to continue. So I try to just illustrate that very simply here. For example, what you have is the original population with some cells here. They're all blue, so they have exactly the same genotype. And then there could be some mutations in some of them. If they don't cause any problem, they will just stay in the population, right? And they will just be there. And then let's imagine that there was an environmental shift or something that changed that put pressure into that population, but it turns out that this cell here that was orange has a much better fitness compared to these cells that are blue. What will happen is over time, under this selection, the population will change so that most of the cells are going to have this orange genotype and fewer cells will have the blue genotype, right? Because the orange ones have an advantage. And that can go on and on and on, and then building these complex structured population structures where you have many, many different mutations, because natural selection can work on them, and natural selection can cause these differences, right? All right, so it's important for evolution. It's also important for normal cell function. For example, this is one example from immune cell, and it's the clonal selection theory. So the immune cells, B cells, are making antibodies, right? And in our environment, we have millions of antigens, right? We have all kinds of different proteins with different surfaces coming from viruses and bacteria, the things we eat, etc., etc. If you had to have an antibody sequence in your genome for millions of different antigens, then our genome would have to be so big, because we can't make proteins, antibodies that are going to be potentially binding to everything on Earth, right? Because the protein surfaces can be very, very different. So how does this problem solve? It's solved by several strategies. So the first strategy that the cells use is they can create a more diverse antibody repertoire by using VDJ recombination. What that means is part of the antibodies are encoded from little different segments in the genome, and there is a mechanism to mix and match these segments, right, to make different antibodies. And there is another mechanism that increases the protein diversity using something called somatic hypermutation. Mutation that occurs in these B cells, lots of mutations, okay? So how does that usually occur? So we have a collection of B cells in our bodies, right? And then when there's an antigen that is recognized by specific B cells, what happens is that those B cells start dividing. And over time, they will divide, and make lots of B cells. And over time, the affinity of these B cells, antibodies to the antigen, increases, right? Just to visualize that. This is how an antibody looks like, right? You have the heavy chain here. In dark blue, you have the light chain here. And the end of the heavy chain and light chain, Y-shaped structure, is the surface that's going to recognize the antigen. Right? So this is a crystal structure where you can see that the complementary determining region, which is here, is basically binding to the antigen. And the stronger that interaction, the stronger the immune response to that antigen, right? And you want that when you want to get rid of some bacteria or virus, etc. So this surface area matures with time. What that means is it gets better and better at recognizing the antigen with time. So the cells make better and better antibodies that have stronger and stronger binding. How that happens? It happens through somatic hypermutation. It's a mutation on purpose. So in B cells, what the cells do is they activate an enzyme called activation-induced deaminase. So this is an enzyme that purposefully goes to that methylated cytosine here and removes an amine group. So deaminates this so it becomes a T, right? And then the cells don't have time to repair this. They get replicated, and then you have a mutation here. So when those mutations occur here, what are they going to change in the antibody? When you have these mutations occurring in the antibody, what are they going to change?
They're going to change the amino acid
Very good.
sequence of the binding region, because of that the structure of the protein is going to change, so they need to become more or less specific, and the ones that are more specific, they're going to be the ones that end up being used against the antigen.
Yeah, you should come here and teach. That was really good. Right? So that you have mutations, it's going to change the amino acid sequence, that's going to change the structure, and that change can either increase the affinity or decrease the affinity. And if it increases the affinity, those cells actually will divide more, and then you're going to have much more cells making the antibodies that are going to be specific, more specific to this antigen. All right. So what would the consequence of hypermutation to most B cells? You're on purpose mutating your own DNA. Right? The cells are on purpose mutating their own DNA. What would be the consequence?
Higher rates of cancer.
That's, I'm not sure about that link. I don't know if that link has been made. What's going to happen to most of the B cells making this mutation? Go ahead.
Most of them are not going to proceed, right?
Most of them are going to die,
Yeah.
right? But some of them are going to survive, and some of them are going to have a higher affinity. Again, coming back to that evolution selection ideas, right? You have a population of cells. Most of them are going to die, some of them are going to survive, and the ones that survive are going to thrive and get more and more in numbers. All right. So that also is a principle that we now use to understand cancers. So you can understand cancers through evolution and ecology. So one of the questions that you might ask, Professor, you told me that mutations can cause the cells to die, and you also told me the cancer cells have a lot of mutations. How are they surviving, right? They have problems with their DNA repair enzymes. They're just fine, right? These cells can grow. And the way to understand that is through, again, evolution. So basically, each cancer cell will accumulate mutations, and some of these mutations will be advantageous for growth, and some of them won't be, right? So those cells will die. So if a cell has mutations that are very advantageous for its growth, it will divide and increase in numbers within the tumor. So the tumor is a population of cells. And then until another cell with an even better advantage starts, right? And then that is going to increase in numbers and will take over the population, etc. So you're going to, in the tumor, you're going to have a population of cells constantly fighting, and whoever has the best fitness wins, right? And that fighting and increasing the fitness occurs through accumulating lots of mutations. All right, same idea, kind of like how evolution over population works, right? What kind of things gives cancer cells advantage? One of the things that can give a cancer cell advantage is if it is able to activate its telomerase again, right? So what the telomerase is basically the enzyme that puts all these repeats in the telomeres, creating these structures. In embryonic cells or stem cells, you have many copies of this repeat, right? So this repeat sequence in TTAGGG. You have thousands of copies of this, giving rise to about 3 to 20 kb of these repeats at the end of the chromosomes. And these chromosomes, the cells with these chromosomes can indefinitely replicate. In somatic cells, so these are our skin cells, liver cells, etc., right? The cells that aren't germ cells. you have a reduced number of these telomeres, repeats, and they are capable of replicating, but a limited number. In senescent cells, these are cells that will no longer divide. You basically have none of these repeats at the telomeres. And so these basically cells live for a while and die. The cancer cells, if you look at them, these guys, somehow they turned on their telomerase and they can make thousands of copies of these repeats again, having a very nice telomere that is capable of supporting indefinite replication again, right? And why does that happen? You've learned DNA replication, and you've learned that due to the lagging, how the lagging strand is synthesized, when you replicate, what happens is that the last portion of the lagging strand is not going to be able to fill in this gap, because the DNA polymerase needs a primer to fill in this gap. What you're going to have is a shortened end in one of the strands, okay? So every time a cell replicates, if there is no telomerase, the ends of the chromosome is going to get shorter and shorter and shorter. Does that make sense? So in the second round of replication of the strand, it's going to get shorter, and it's going to get shorter, it's going to get shorter. So every time the cell divides, it's going to produce progeny with shorter and shorter ends. Okay? In cells that divide a lot, this problem is solved by this complex called the telomerase. It's an RNA protein complex, and what it does is it uses this RNA, which has the same sequence that has that telomere repeat sequence, right, as a template to synthesize more DNA, right? So what you have is you have the telomerase enzyme with the RNA, and what it does is it adds this DNA to the complementary strand, and adding in more and more. And then once it adds the whole repeat sequence, it moves on a little bit. It slides in a little bit and then starts the process, so adding and increasing the length, right? And once it increases enough, so once this strand is increased enough, right, it can serve as another site for the lagging strand to start with a new primer, right? And then that's going to fill this in, and you're going to have a longer end, right? So the telomerase will increase the end of the chromosome. Does that make sense? Any questions? All right. So what enzymatic activity should be in the telomerase? What is it doing? It's adding DNA with an RNA template. Reverse transcriptase, right? So it has to have a reverse transcriptase activity. It's another question. Which Marvel character likely has lots of telomerase? Wolverine, my favorite, right? Because it's constantly regenerating. Yeah? Oh my God, thanks for going there. That is really good. I should add that. Definitely. It's a good point. All right. So the cancer cells basically have telomerase to be able to divide indefinitely, right? And one of these cells that have been dividing indefinitely are the first cells that were isolated to support in vitro culturing of human cells. And these cells were isolated from a person named Henrietta Lacks. And you might have heard her name because it belongs to the cells, this cell line called HeLa cells, okay? Henrietta Lacks cells. So she had ovarian cancer, and in Johns Hopkins, the researchers who were trying to develop these tissue culture systems to grow human cells found out that her cancer cells were supporting growth a lot. So they were dividing like crazy. They were really happy in the dish. And here's a picture of these HeLa cells under a microscope using immunofluorescence. All right, some quick facts about HeLa cells. The amount of HeLa cells that were grown across the world, because they're now sent all over the world, are about 20 tons. So about 20 tons of HeLa cells have been grown. There's about 300 papers per month that use HeLa cells for their experiments. And in fact, Henrietta Lacks actually died quite young. So her cells lived outside her body longer than they actually lived inside her body. Okay? So, and there's a really nice book by Rebecca that is titled The Immortal Life of Henrietta Lacks, that tells you the story of these cells and the story of the family who are the Lacks family. And I think they turned it into a movie as well. All right, so how do these cells actually look like? So they've been distributed to all the labs across the world, and these cells have basically adapted to lab condition. They can live, they can divide a lot in a dish, right? So if you then look at the karyotype of these cells, it is amazing that they look very, very different from a normal karyotype, right? So this is the normal karyotype of the male, and this is the HeLa karyotype. What you can see is that there are 3 copies of chromosome 1 now. There's 1 copy of chromosome 2. There's like all these chromosomes where you can't even call them a certain chromosome because they've been spliced and translocated to each other, completely jumbled up, right? And then there's some chromosome parts missing because those genes have been kind of translocated with other chromosomes, etc. So what you can see is that here's a karyotype that has evolved for living in a dish, right? And that's pretty amazing. All right. So here's a link to a helpful animation, and I'll see you Monday.